Lab 24, Protein Synthesis. Introduction. RNA is produced from DNA through a process called transcription. RNA molecules are transferred according to the information encoded in the base sequence of DNA. An enzyme called RNA polymerase first binds to a DNA molecule and then causes the separation of the complementary strands of DNA. The enzyme next directs the formation of hydrogen bonds between the bases of a DNA strand and complementary bases of RNA nucleotides that are floating in the nucleus. RNA polymerase then moves along the section of DNA, establishing the sugar-to-phosphate bonds between the RNA nucleotides in much the same way as in DNA replication. When RNA polymerase reaches the sequence of bases on the DNA that acts as a termination signal, the enzyme triggers the release of the newly made RNA. All three types of RNA are transcribed in this manner. Each type of RNA then leaves the nucleus and travels to the cytoplasm, where it is involved in the synthesis of protein. The transcription process creates RNA with a base sequence complementary to DNA. During transcription, the genetic code of DNA becomes inherent in the sequence of bases in RNA. The process of assembling protein molecules from information encoded in mRNA is called translation. The process of translation begins when mRNA moves out of the nucleus by passing through the nuclear membrane. The mRNA then migrates to a group of ribosomes where the actual synthesis of protein takes place. tRNA transforms amino acids floating freely in the cytoplasm to the ribosomes. At the top of each tRNA, there's a region that bonds to the specific amino acid. At the opposite end of the clover-like structure, tRNA bears a sequence of three bases called an anticodon. The tRNA anticodon bases are complementary to and thus pair with the codon of mRNA. The assembly of a protein begins when a ribosome attaches to an AUG codon on the mRNA, and the AUG codon pairs with an anticodon UAC on a specific tRNA. Once the tRNA with this anticodon carries methionine, the first amino acid in the chain is always methionine. There are usually several ribosomes simultaneously translating the same mRNA. As a ribosome moves along the strand of mRNA, each codon is sequentially paired with its anticodon, and the specified amino acid is added to the chain. An enzyme in the ribosome catalyzes a reaction that binds each new amino acid to the chain. In this reaction, a chemical bond is formed with the last amino acid in the chain. The process continues with each amino acid being linked to the chain in turn. Eventually, the ribosome reaches a stop codon. This codon brings the process to an end. At this point, the mRNA is released and the protein is complete. In this activity, you will simulate the construct of a model of DNA and determine the amino acids in a protein coded for in the base sequences of the DNA. Procedure 1. Obtain 2 sheets of blank white paper and tape them together. 2. Obtain the sheet of paper containing DNA nitrogen bases. This represents the 2 halves of a DNA molecule. 3. Cut out the DNA sequence and tape it onto one long strip by attaching strip 2 to the end of strip 1. 4. The bottom of the strip is the template for synthesizing the mRNA strand. 5. Tape the strip to the top of your white paper. Obtain the mRNA codons. Cut them out and transcribe them from the DNA strand. Place Paste them beneath the DNA strand, leaving some space between the DNA and the mRNA. 7. Cut out the tRNA. Align the appropriate tRNA anticodon below the appropriate mRNA codon. Glue the tRNA down to your paper in this position. 8. Next, align the appropriate amino acid next to the appropriate tRNA. Use the genetic code to translate the mRNA strand into a sequence of amino acids. 9. Continue the process until a stop codon is reached. 10. Label your diagram using all appropriate labels.